Hi, I am Susanne Paser. I am from the Institute of Physiological Chemistry, Department of Biochemical Endocrinology at the University of Duisburg-Essen in Germany.
Hi, I am Daniel Sebastian Dohle. I am from the same institution as Susanne.
Hi, I am Sebastian Mustmann from the Institute for Anatomy, Department of Neuroanatomy at the University of Duisburg-Essen.
We are going to show you how chick ex ovo culture and chorioallantoic membrane assay really work. So let's go to the lab.
In most of the recent experimental studies, the CAM, that is, the chick chorioallantoic membrane, was exposed by cutting a window through the eggshell, and experiments were carried out in ovo, resulting in significant limitations in the accessibility of the CAM and in the possibilities for observation and photodocumentation of effects. Ex ovo cultures of chick embryos significantly facilitate the accessibility of the CAM and the chick embryo, please see the beating heart, enabling easy in vivo documentation of effects and experimental manipulation of the embryo. In our video article, we are going to provide a step-by-step demonstration of the successful application of ex ovo culture for a large number of scientific questions. The eggs are incubated in an incubator with a moving tray. Here you can see the heating element and the tray mover rotating the eggs 12 times a day continuously. The humidity is kept at 60% by adding water to plastic containers at the bottom of the incubator, and the incubation temperature is set at 37.5 degrees centigrade. Before starting incubation, dirt, feathers, and excrements are carefully removed from the eggshells mechanically. Wiping the eggs with ethanol or disinfectant, however, significantly reduce the survival rates of the embryos. The eggs are positioned horizontally on the moving tray of the incubator. Please make sure to keep enough distance between the individual eggs to enable free rotation. After incubation for 72 hours, the eggs are removed from the incubator, and the top side of the eggs where the embryo resides is marked by a pencil, since the embryo resists rotation to a certain extent and remains located here. For a better feeling, no gloves should be used to crack the eggs, but the hands should be disinfected with 70% ethanol. The egg is held horizontally with a pencil mark on top and cracked on the edge of a triangular magnetic stirring bar. For maximum force control during the cracking procedures, the elbows should not rest on the bench top. It is important that the eggshell as well as the egg membrane are opened during cracking. Leaking of egg white is a safe sign that the egg membrane has been perforated. The egg is kept close to the bottom of the petri dish to avoid further leakage of egg white. During this initial opening procedure, it is important that the egg white on the bottom of the petri dish is still connected to the rest inside of the egg, as this results in a vacuum inside the egg, holding the yolk in place. By gently applying pressure to the meridian running through the crack and the equator of the egg by thumb and middle fingers, it is possible to regulate the vacuum and the extrusion of the egg contents. The content of the egg can then be transferred to the petri dish with the yolk undamaged if the egg is gently lifted and both halves of the shell are carefully separated. The ex ovo cultures are then placed on a tray, returned to an incubator, and kept at 37.5 to 38.2 degrees centigrade and 70% humidity. A carbon dioxide or oxygen supply is not necessary if special petri dishes with spacers between lid and dish and incubators with a ventilation grid are used. The ventilation grid should be kept halfway open. Autoclaved filter papers are used as supporting material for liquid substances applied to the cam, as they seem to cause the least irritation of the cam. The application site should be halfway between the embryo and the border of the cam, as otherwise the embryo hinders the microscopic observation of effects in transmitted light. The liquid, here angiotropin, should be applied immediately after the filter has been placed onto the cam. To avoid drying, either additional doses or buffer should be reapplied every day. When the cam is fully developed, up to 6 different samples may be applied and their effects can be compared on a single cam. Rings cut from a 1 milliliter pipette tip should be cut as thin as possible to minimize weight and to avoid indentation of the cam. The ring is then applied to the cam and the cells are pipetted into the ring. A Hamilton microliter syringe is rinsed several times with 70% ethanol and finally with sterile phosphate buffered saline, or PBS. The microsyringe is filled with the prepared cell suspension. Carefully but quickly penetrate the cam and the eye layers with the syringe needle and continuously inject the cell suspension. The needle should remain some seconds in the eye to avoid a loss of the injected cell suspension by leakage. Chick ex ovo cultures of eggs incubated for 3 days are used as limb bud donors. The embryo is dissected from the connected yolk vessels by cutting a circle. The embryo is transferred to a petri dish filled with cold, sterile PBS by use of a small drained spoon and washed by agitation. The embryo is then transferred to a fresh petri dish filled with sterile PBS and freed from the surrounding membranes, carefully tearing them apart with a fine forceps. The limb buds are detached by forceps as close to the body as possible, grasped and transferred to the cam of an 8 to 10 day old host chicken. At the desired application site, the cam is selectively traumatized by gentle scraping with one chunk of the forceps. Then the graft is grasped and layered onto the cam with the other chunk of the forceps. The sacrificed female mouse is fixed on a board. The abdominal wall is moistened with 70% ethanol, cut along the midline, and the skin flaps are fixed laterally by pins. The uteruses are removed from the abdomen, detached, and transferred to a beaker with cold PBS. The embryonic membranes are removed carefully by the use of forceps. For demonstration purposes, a 16-day-old mouse embryo is shown here for better visibility of the limb buds. Optimal grafting results are only obtained, however, if limb buds from 10 to 13-day-old embryos are used. The limb buds are cut off by the use of fine forceps as close to the body as possible. At the desired application site, the cam is selectively traumatized by gentle scraping with forceps. Then, the limb buds are grasped and transferred to the cam of an 8 to 10-day-old host chicken. The graft is placed once or twice on a side of the cam where no final grafting is intended to remove excess PBS. The grafts are ideally placed on the cam near a Y bifurcation of a blood vessel. Tumor biopsies are cut into 1 to 2 cubic millimeter pieces by sterile scalpels. Taking material from the surface of the biopsy increases the risk of contamination with muscle or connective tissue. At the desired application site, the cam is selectively traumatized by gentle scraping. A piece from the core of the biopsy sample is grafted onto the cam of an 8 to 10-day-old host chicken. The grafts are ideally placed on the cam near a Y bifurcation of a blood vessel. The graft is then transferred to the cam with minimal PBS attached. Light pressure might be applied to maneuver the graft into a resulting indentation of the cam. The host chick embryo is killed by decapitation. The cam with the attached graft is removed by a circular cut and transferred to a petri dish filled with PBS. Excessive cam is cut from the graft except for the former attachment site. Again, at the desired application site, the cam of a new host chick embryo is selectively traumatized by gentle scraping, and the graft is transferred to the cam of the second host with minimal PBS attached.
We just demonstrated how chick ex-ovo culture really works, showed you the advantages compared to in-ovo experiments, and gave you some examples for its application. So that's it. Thank you for watching and good luck with your experiments.